Every breath you take. Yeah, every single beat of your heart, even every thought you have, it all comes down to one thing, really. Your cells needing oxygen.
It's a huge demand, constantly.
Absolutely. And it's met by this, well, incredible cellular machinery.
Truly amazing stuff.
So today, we're diving deep. We're looking into a really fascinating chapter from Karp's Cell and Molecular Biology, the 8th edition.
A good one. Yeah, that chapter really unpacks aerobic respiration and the mitochondrion.
Exactly, the mighty mitochondrion. And our mission here is basically to break down this complex science. Make it clear, exciting. We'll hit the core concepts, how they figured this stuff out experimentally, and importantly, why it matters to you listening.
Right. There are definitely some surprising facts in there.
Oh yeah, get ready for some aha moments.
Okay, so to really get why mitochondria are so vital, we kind of have to go way back, billions of years back.
Billions, okay.
Yeah. Think about early Earth's atmosphere. It was nothing like today. It was full of what we call reduced molecules, like hydrogen gas. Life back then, entirely anaerobic.
Meaning no oxygen needed.
Precisely. Organisms got by with things like glycolysis, maybe fermentation, processes that honestly only capture a tiny bit of the energy available in food molecules.
Right, not very efficient.
Not at all compared to what came later. Then, maybe 2.7 billion years ago, something huge happened. Cyanobacteria showed up.
Ah, the blue-green algae guys.
Those are the ones. They developed this new kind of photosynthesis that split water molecules, and the byproduct, oxygen, molecular oxygen.
So they started pumping oxygen into the atmosphere.
Exactly, and it started accumulating. This completely changed the planet. Oxygen was actually pretty toxic to a lot of early life, but it was also this powerful force for natural selection.
Survival of the fittest, oxygen style.
You could say that. Organisms evolved not just to tolerate oxygen, but to actually use it. Aerobes emerged, and they could fully oxidize compounds down to CO2 and water, releasing way, way more energy.
A huge evolutionary advantage.
Massive. This basically paved the way for all the complex oxygen-breathing life we see today, including us.
Okay, and here's where it gets really wild, right? The mitochondria connection.
This is the mind-bending part. The place where this oxygen utilization happens in our cells, in your cells, is the mitochondrion.
Yeah.
But mitochondria weren't originally ours. The evidence points strongly to them originating as free-living bacteria.
Bacteria.
Yeah.
Living inside our ancestor cells.
Exactly. They took up residence inside some primitive host cell billions of years ago, endosymbiosis. So that oxygen you're breathing in right now, it's being consumed, literally used up by these tiny descendants of bacteria living inside your cells.
Wow. So we're breathing for our internal bacteria buddies.
Oh,
that's something.
It's a direct link back to the dawn of complex life, a profound evolutionary legacy.
Okay, so tell us more about these. Former bacteria. What are mitochondria like? Are they just little static beans?
Not at all. They're actually quite dynamic. You can see them with a regular light microscope. They're big enough for that. But their shape varies hugely. Sometimes they are sort of bean-shaped, maybe 1 to 4 micrometers long. But they can also form these long, branched, interconnected networks. And they're constantly changing shape. They can fuse together.
Merge.
Yeah, merge into larger structures. Or they can split in two, undergoing fission.
And how does that splitting happen?
Well, this is really surprising. Often it's triggered by contact with the endoplasmic reticulum, the ER.
The ER. How?
Thin tubules of the ER can actually wrap around a mitochondrion, almost like a noose, and initiate the constriction that leads to splitting.
That's wild. So there's this constant balancing act.
Exactly. The balance between this fusion and fission controls how many mitochondria a cell has, their overall shape, how connected they are, and it's critical. Mutations in the proteins that control mitochondrial fusion are actually linked to some serious inherited neurological diseases.
Wow. Okay, so they're dynamic, they interact with other organelles. What else? Are they just about energy?
Well, energy is the main job, their claim to fame. They're incredibly numerous, making up maybe 15–20% of the volume of a liver cell, packed with over 1,000 different proteins.
A significant chunk of the cell.
Absolutely. And yeah, they're often found snuggled up next to fatty acid droplets. Those droplets are like their fuel supply.
Makes sense.
A really striking example is in sperm cells. The midpiece is just crammed with mitochondria, powering that tail whipping back and forth. And of course, plant cells rely on them too for ATP.
But you mentioned other roles.
Yeah.
Beyond ATP.
Right. They're involved in making other essential molecules like certain amino acids and heme groups for proteins like hemoglobin. They also work with the ER to manage calcium levels in the cytosol, which is vital for cell signaling.
Calcium regulation.
And a really big one, they play a crucial role in regulating programmed cell death or apoptosis.
Cell suicide pathways. Important stuff. Okay, so that's the big picture. Let's dive into the structure. You said two membranes.
Two distinct membranes, yes. There's the outer membrane, which smoothly covers the whole organelle. It's surprisingly permeable.
Let things through easily.
Relatively speaking, yes, thanks to channel proteins called porins. And interestingly, these porins look a lot like proteins found in the outer membranes of bacteria.
Another nod to their origins.
Exactly. Then you have the inner membrane. And this one is very different. It's highly impermeable.
The opposite of the outer one.
Right. It's jam-packed with proteins over 100 different kinds. The protein-to-lipid ratio is way higher, something like 3:1. It doesn't have cholesterol. And it's rich in this unique phospholipid called cardiolipin.
Which is also found where?
In bacterial plasma membranes. Another link.
Okay, so this inner membrane is impermeable and protein-rich. What about its shape?
It's not just a simple bag inside the outer one. It's extensively folded. These folds are called cristae.
Cristae. I remember those from biology class. They increase the surface area, right?
Massively. And that's crucial because the cristae are where the action happens. They house all the molecular machinery for aerobic respiration and making ATP.
The power generators are built into these folds.
Precisely. They're highly organized, connected to the main part of the inner membrane by narrow tubular sections called cristae junctions. There's even a specific protein complex called mitose or mitose that helps maintain this intricate folded structure.
Okay, two membranes fold. What about the spaces they create?
Right, the membranes define two separate aqueous compartments. There's the inner membrane space between the outer and inner membranes, and then there's the innermost compartment, the matrix.
The matrix? What's in there?
It's like a dense, gel-like substance packed with soluble proteins. It also contains mitochondrial ribosomes.
Ribosomes, like the cell's protein factories.
Their own version, yes. And crucially, several copies of circular mitochondrial DNA.
Circular DNA, just like bacteria.
Exactly. It's a remnant, a relic of that ancient bacterial ancestor. In humans, this mitochondrial DNA, or mtDNA, is quite small. It only codes for 13 polypeptides, plus a couple of ribosomal RNAs and 22 transfer RNAs needed to make those proteins inside the mitochondrion.
Only 13 proteins. Where did all the other bacterial genes go?
Good question. Over evolutionary time, most were either lost or, more commonly, transferred to the host cell's nucleus. So most mitochondrial proteins are actually encoded by nuclear genes, made in the cytosol, and then imported.
Fascinating. And this mtDNA is useful for tracing ancestry.
Very much so, because it's inherited almost exclusively from the mother via the egg, and because it tends to mutate faster than nuclear DNA, it's been a hugely valuable tool for tracking human migration patterns and evolutionary history.
Amazing. Okay, structure, origins, check. Now let's get to the main event, the energy production. How does the mitochondrion actually squeeze energy out of food molecules like glucose?
Right. It starts outside the mitochondrion in the cytosol with glycolysis.
Initial breakdown of sugar.
Exactly. Glucose gets split, goes through a series of steps, but glycolysis itself only yields a tiny net gain of energy, just 2 ATP molecules per glucose. Most of the energy from that glucose is still locked up in the end products, mainly pyruvate and also in some NADH molecules.
Okay, so glycolysis is just the appetite.
A very small appetizer. But if oxygen is around, things get much more interesting. That pyruvate doesn't just hang around, it gets transported into the mitochondrial matrix.
Across both membranes.
Into the matrix, yes. Once inside, it undergoes a key reaction. It gets decarboxylated, meaning a carbon atom is removed as CO2, and the remaining 2 carbon unit, an acetyl group, gets attached to coenzyme A. Forming acetyl-CoA. This reaction is carried out by a huge multi-enzyme complex called pyruvate dehydrogenase.
Acetyl-CoA. So that's the key molecule that enters the next stage.
That's the one. Acetyl-CoA is the primary fuel for the next big pathway, the tricarboxylic acid cycle, or TCA cycle.
Also known as the Krebs cycle, or citric acid cycle.
All the same thing. And most of the enzymes for this cycle are right there in the mitochondrial matrix.
Okay, what happens in the TCA cycle?
Acetyl-CoA comes in and merges with a 4-carbon molecule called oxaloacetate. This forms a 6-carbon molecule, citrate, hence the name citric acid cycle. Then, through a series of enzyme-catalyzed steps, that citrate is progressively oxidized. 2 carbons are released as CO2, and crucially, oxaloacetate is regenerated at the end.
So the cycle can keep turning as long as there's acetyl-CoA coming in.
Precisely. It's a true cycle. Now, the CO2 is waste, But the really important products are the reduced coenzymes.
NADH and FADH2.
Yes, for each turn of the cycle, processing one acetyl-CoA, you get 3 molecules of NADH and 1 molecule of FADH2. These molecules are carrying high-energy electrons. That's their key role.
Electron carriers. Got it.
Yeah.
Any direct ATP.
Well, 1 molecule of GTP, which is energetically pretty much the same as ATP, is also produced per cycle turn. But the main energy payoff comes from those electron carriers.
And this cycle isn't just for glucose breakdown.
Not at all. That's why it's often called a central metabolic hub. The breakdown products of fatty acids get broken down into acetyl-CoA, and proteins, amino acids, can be converted into various intermediates of the cycle. They all feed into the TCA cycle.
So it's a convergence point for oxidizing all sorts of fuel.
Exactly. It processes the carbon skeletons derived from pretty much all energy-providing macromolecules.
Okay, so we have NADH and FADH2 loaded with high-energy electrons inside the matrix. What about the NADH made outside during glycolysis? How does its energy get used?
Good point. The inner mitochondrial membrane is impermeable to NADH. So the NADH generated in the cytosol can't just wander in.
So how do those electrons get inside?
Through clever shuttle systems. There are a couple of main ones. The malonaspartate shuttle is one. It effectively passes the electrons from cytosolic NADH onto NAD plus inside the matrix, regenerating NADH inside.
Okay, so the electrons get in, carried by different molecules.
Right. The other major one is the glycerol phosphate shuttle. This one transfers electrons from cytosolic NADH to FAD that's actually part of an enzyme embedded in the inner mitochondrial membrane, producing FADH2 right there.
So both shuttles funnel electrons into the mitochondrial energy production line.
Correct. They both deliver electrons to the next stage, the electron transport chain, setting the scene for the big ATP payout. And that brings us to oxidative phosphorylation. This is where the vast majority of ATP is generated from the energy stored in those electrons carried by NADH and FADH2.
Oxidative phosphorylation sounds important. 2 main steps, you said.
2 key phases, yeah. First, those high-energy electrons from NADH and FADH2 are passed along a series of protein complexes embedded in the inner mitochondrial membrane. This is the electron transport chain.
Like a bucket brigade for electrons.
Sort of, yeah. As the electrons move from one carrier to the next, they release energy. And this released energy is used for something crucial: pumping protons, H+ ions, from the matrix out into the intermembrane space.
Pumping protons against their concentration gradient.
Exactly. It builds up a higher concentration of protons in the intermembrane space. This creates an electrochemical gradient. And what happens to the electrons at the end of the chain?
They have to go somewhere.
They're transferred to the final electron acceptor, molecular oxygen. Oxygen gets reduced, combining with protons to form water.
So that's where the oxygen we breathe actually gets used, to accept those electrons.
That's its critical role. Without oxygen, the chain backs up and energy production grinds to a halt. Okay, so that's step 1. Electron transport coupled to proton pumping.
Creating a proton gradient. What's step 2?
Step 2 harnesses the energy stored in that gradient. The protons that were pumped out now flow back into the matrix down their electrochemical gradient. But they don't just diffuse back randomly.
They go through a specific channel.
Precisely. They flow back through a remarkable enzyme complex embedded in the inner membrane called APP synthase. And the energy released by this downhill flow of protons is used by ATP synthase to attach a phosphate group to ADP, making ATP.
Wow. So the proton flow powers ADP synthesis.
That's the essence of it. This whole idea linking electron transport, proton pumping, and ATP synthesis via proton gradient is the chemiosmotic mechanism. Peter Mitchell proposed it back in 1961, a Nobel Prize-winning insight.
Chemiosmosis. Okay. So how do we actually measure this energy release? How do we know electrons are flowing downhill?
We use a concept called oxidation reduction potential, or redox potential. It's basically a measure of a substance's affinity for electrons, measured in volts.
Higher potential means?
Higher potential means a stronger tendency to accept electrons, a stronger oxidizing agent. Lower potential means a stronger tendency to donate electrons, a stronger reducing agent.
Okay.
The difference in redox potential between an electron donor and an electron acceptor tells you how much free energy is released when electrons move between them. For example, NADH is a strong reducing agent, very low redox potential. Oxygen is a very strong oxidizing agent, very high redox potential.
So the transfer from NADH to oxygen
releases a large amount of energy. It's a very favorable downhill process energetically. And the electron transport chain captures this energy release in steps.
makes sense. And what are the actual components of this chain, these electron carriers?
They're a diverse group, mostly proteins with specific non-protein components called prosthetic groups that handle the electrons. Except for one, ubiquinone, which is lipid-soluble.
Okay, what kinds are there?
Well, you have flavor proteins that contain flavin groups derived from riboflavin, cytochromes, which contain iron in a heme group, similar to hemoglobin, copper atoms associated with certain complexes, iron-sulfur proteins, where iron is linked to inorganic sulfide ions, and that ubiquinone or coenzyme Q, which is small and mobile within the membrane.
A whole team of specialized electron handlers.
Exactly. And they are arranged within the inner membrane in a specific sequence, generally in order of increasing redox potential.
So electrons naturally flow from lower to higher potential carriers.
Correct, releasing energy at each major step, ultimately ending up on oxygen. Scientists actually figured out this sequence back in the day using specific inhibitors.
How did that work?
They'd use a poison, say rotenone, which blocks electron transfer at a specific point early in the chain. Then they'd look at the carriers before the block. They'd all be stuck in their reduced state full of electrons. The carriers after the block would be oxidized, empty. By using different inhibitors blocking different steps, they could piece together the entire sequence.
Clever detective work. Okay, so these individual carriers, they're grouped into larger structures, right? The complexes.
Yes, the major carriers are organized into 4 large multi-protein complexes, numbered I through 4, that span the inner mitochondrial membrane.
Complex 1, 2, 3, and 4.
Right. And ubiquinone and cytochrome C act as mobile shuttles carrying electrons between these large fixed complexes.
Okay, let's walk through them. Complex 1.
Complex 1, also called NADH dehydrogenase. This is the entry point for electrons coming from NADH generated in the matrix, like from the TCA cycle. It takes electrons from NADH and passes them to ubiquinone. And critically, complex 1 is a proton pump.
Pumping protons out. You mentioned it's linked to Parkinson's.
Yes, dysfunction in complex Psi has been strongly implicated in Parkinson's disease, possibly due to environmental toxins or genetic factors. Now, complex 2 is different.
How so?
Complex 2, or succinate dehydrogenase, is actually also an enzyme in the TCA cycle. It's the entry point for electrons from FADH2. It passes these electrons also to ubiquinone. But importantly, complex 2 does not pump protons.
Okay, so FAEH2 electrons contribute less to the proton gradient.
Exactly, because they bypass the first proton pumping site at complex 1. From ubiquinone, electrons move to complex 3, the cytochrome BC1 complex.
What does the 3 do?
It takes electrons from reduced ubiquinol, and passes them to the next mobile carrier, cytochrome C. And complex 3 is a proton pump. It pumps about 4 protons across the membrane for each pair of electrons that pass through it.
Pumping more protons, then where do electrons go from cytochrome C?
To the final complex, complex 4, also known as cytochrome C oxidase. This is the grand finale.
Yeah, oxygen step.
The oxygen step. Complex 4b takes electrons, one at a time, from 4 cytochrome C molecules and transfers them to a molecule of molecular oxygen, O2. This reduces the oxygen, which combines with protons from the matrix to form 2 molecules of water.
H2O, wow.
And complex 4 is also a crucial proton pump. For every oxygen molecule it reduces, it consumes 4 protons from the matrix to make water, but it also actively pumps another 4 protons across the membrane.
So it contributes significantly to the proton gradient too.
Very much so. And this process is incredibly efficient. The cell manages to transfer 4 electrons to oxygen without releasing dangerous, partially reduced oxygen intermediates.
Like superoxide radicals.
Exactly. Complex 4 holds on to the oxygen until it's fully reduced to water. That efficiency is vital. It's also the site where poisons like carbon monoxide, azide, and cyanide bind, shutting down respiration.
That's why they're so deadly.
Precisely. They block the final step. Scientists even proved complex 4V pumps protons by isolating it, putting it into artificial membrane vesicles, and showing that when it processed electrons, protons were indeed pumped out of the vesicles.
Direct experimental proof. Okay, so we built up this huge proton gradient, this proton motive force. Let's connect it directly to making ATP. How does that force get used?
Right, the proton motive force, or APOLA. It's the potential energy stored in that gradient across the inner membrane. It has 2 components.
How do they?
There's a chemical component, the difference in proton concentration, the pH gradient. It's more acidic in the inner membrane space. And there's an electrical component, a voltage across the membrane, because you're pumping positive charges protons out, making the outside positive relative to the matrix.
So concentration difference and electrical difference.
Yes. In our mitochondria, the electrical voltage part actually accounts for most of the energy, maybe 80% of the total proton motive force. You can even see this potential using special fluorescent dyes.
Cool. Now, what happens if this process gets uncoupled?
Uncoupling. That means electron transport continues, oxygen is consumed, but ATP isn't made because the proton gradient is dissipated without going through ATP synthase. The energy just gets released as heat.
Like with that old diet drug DNP.
Exactly.
Yeah.
Dinitrophenol basically acted like a proton shuttle, making the inner membrane leaky to protons. People burned calories like mad, generated a lot of heat, but couldn't make enough ATP to survive. Very dangerous.
But there are natural uncouplers, too.
Yes, there are uncoupling proteins, UCPs. The most famous is UCP1, found in brown adipose tissue or brown fat.
Brown fat for heat generation.
Precisely. It's important for keeping infants warm, and we now know adults retain some functional brown fat, too. UCP1 essentially provides a regulated pathway for protons to leak back into the matrix, generating heat instead of ATP. Other UCPs might exist to prevent the proton gradient from getting too large, which could otherwise lead to excessive production of damaging reactive oxygen species, or ROS.
A protective mechanism, maybe. Okay, so assuming things are coupled, let's meet the machine that does the coupling, ATP synthase.
Star of the show, in many ways. ATP synthase is this large, absolutely amazing protein complex. Looks kind of like a mushroom sticking into the matrix.
A mushroom. Okay. Two main parts.
Two main parts. There's the big spherical F1 head, which protrudes into the matrix. This is where the actual catalytic sites for making ATP are located. Then there's the faux base. The O is for oligomycin, a drug that blocks it, which is embedded within the inner mitochondrial membrane. This part contains the channel that protons flow through.
And they're connected.
Yes, by a central stalk and also a peripheral stalk. It's quite an intricate structure. When researchers first isolated just the F1 head, they found it acted as an ATPase. It broke down ATP.
Which seems backward, right?
It did initially.
Yeah.
But enzymes catalyze reactions in both directions. It turns out the enzyme can run in reverse. And experiments with other membrane pumps showed that if you create a steep ion gradient, you can actually force them to run backward and synthesize ADP. It demonstrated the principle. Ion gradients can drive ATP synthesis.
Okay, so the proton flow through Fo drives ATP synthesis in F1. But how? This is the binding change mechanism, right? Paul Boyer's idea.
Exactly, and it's brilliant. Boyer proposed that the energy from the protons flowing down their gradient isn't used directly to force ADP and phosphate together. That reaction actually happens more or less spontaneously once they're bound correctly in the active site.
So what is the energy used for?
The energy is used to change the shape of the catalytic site, specifically to release the newly formed ATP molecule, which binds very tightly.
Release the ATP. Okay. How does that work?
The F1 head has 3 catalytic sites located on its beta subunits. Boyer proposed these sites cycle through 3 distinct conformations, or shapes. Loose, L, where ADP and phosphate bind loosely. Tight, T, where they bind tightly and ATP is spontaneously formed, and open, O, which has a very low affinity, allowing the finished ATP molecule to be released.
LTO, loose, tight, open.
Right. And the key insight was rotational catalysis. The central stalk connected to the faux part actually rotates inside the stationary F1 head.
It spins.
It literally spins, and as the central stalk rotates, driven by the proton flow through faux, it pushes against the inside surfaces of the beta subunits, forcing them to change shape sequentially through the L, T, and O states.
So electrical energy from the proton gradient becomes mechanical energy rotation, which then becomes chemical energy stored in ATP.
That's the essence of it. It's an energy transduction marvel.
A spinning molecular motor. That sounds almost like science fiction. Is there solid proof for this rotation?
Oh, absolutely fantastic proof. First, structural biology. John Walker determined the atomic structure of the F1 head in 1994, and guess what? The 3 catalytic sites were caught in different conformations, 1L, 1T, 1O, exactly as Boyer predicted. The structure also showed how the central gamma subunit was positioned to interact differently with each beta subunit upon rotation.
Okay, structural evidence. What about seeing it move?
That came next in 1997 from Masasuke Yoshida's lab. This was incredible. They attached a long, fluorescently labeled actin filament visible under a microscope to the end of the central gamma stalk of an isolated F1 unit.
Like a tiny propeller blade.
Exactly. Then they fed it ATP, making the enzyme run in reverse, hydrolyzing ATP, and they watched. And the actin filament rotated counterclockwise, powered by ATP breakdown. They could even see it pause in 120-degree steps at very low ATP concentrations.
They actually saw the motor spin?
They saw it spin. It was definitive proof of rotation. Later experiments even went further. They attached a magnetic bead, forced the stalk to rotate using magnetic fields, and showed that this forced rotation synthesized ATP.
Wow. Direct cause and effect.
Absolutely. It confirmed ATP synthase is a rotary motor, a true nanomachine. Along with bacterial flagella, it's one of the very few examples of rotating machinery in biology.
And the faux part, the base, is what drives this rotation using protons.
Yes. The faux base contains a ring of multiple identical subunits called C subunits. This C ring is attached to the central gamma stalk and sits within the membrane. Protons flowing from the inner membrane space through a channel involving the A subunit of faux bind to an acidic residue on one of the C subunits. Binding causes that C subunit, and thus the whole ring, to rotate slightly relative to the stationary A subunit. The proton rides along until it reaches another channel facing the matrix, where the lower proton concentration causes it to unbind and exit into the matrix.
Like a revolving door powered by protons.
A perfect analogy. And as the C-ring rotates, driven by the proton flow, it forces the attached gamma stalk to rotate along with it, driving the conformational changes in ATP synthesis in the F1 head above.
Incredible mechanism. So making ATP is the main job powered by the proton gradient, but are there other things it does?
Yes, that proton motive force is used for other transport processes across the inner membrane too. It's needed to actively transport ADP into the matrix and ATP out into the cytosol. It also drives the import of inorganic phosphate needed for ATP synthesis.
So getting the ingredients in and the product out.
Right, and it powers other things too, like calcium ion uptake into the mitochondria, some aspects of mitochondrial fusion, and the import of those thousands of nuclear-encoded mitochondrial proteins we talked about earlier.
A very versatile energy source. Yeah. Okay, let's switch gears slightly and talk about peroxisomes. Related, but different.
Related in some ways, yes, but definitely distinct organelles. Peroxisomes are simpler, basically single membrane vesicles. They vary in size, and sometimes you can see a dense crystalline core inside, which is packed with enzymes.
And what do they do?
A whole range of things, actually. They contain over 50 different enzymes. One key role is oxidizing certain molecules, particularly very long-chain fatty acids, ones that mitochondria don't handle well. They're also essential for synthesizing plasmalogens.
Plasmalogens?
They're a special class of phospholipids, really abundant in the myelin sheaths around nerve cells. Problems with making plasmalogens cause serious neurological issues. Fun fact, the enzyme luciferase, which makes fireflies glow, is also found in peroxisomes.
Oh, okay. How are they similar to mitochondria?
Well, both typically arise by division from pre-existing organelles, and they even share some proteins involved in this division process. Both import almost all their proteins from the cytosol after they're made, and both carry out oxidative metabolism, breaking down molecules. There's even evidence now that mitochondria can butt off vesicles carrying specific cargo for peroxisomes.
So they cooperate. What about the name peroxisome?
That comes from hydrogen peroxide, H2O2. Peroxisomes generate hydrogen peroxide as a byproduct of some of their oxidative reactions.
Which is toxic stuff, right? Reactive.
Very reactive, yes. But the peroxisome is uniquely equipped to deal with it. It contains huge concentrations of an enzyme called catalase, which rapidly breaks down the hydrogen peroxide into harmless water and oxygen. It generates the toxin and immediately neutralizes it.
New trick. Any specialized types?
In plant seedlings, yes. They have specialized peroxisomes called glyoxysomes. These contain enzymes for the glyoxylate cycle, which allows the seedling to convert stored fats into carbohydrates, essential fuel for growth, before photosynthesis kicks in.
Okay, interesting organelles. Now let's bring this back to us. The human perspective. What happens when these powerhouses, mitochondria or peroxisomes, don't work correctly?
This is where the clinical relevance really hits home. Mitochondrial diseases are a diverse and often devastating group of disorders.
Affecting which tissues most?
Typically tissues with very high energy demands, like skeletal muscle, heart muscle, and the brain. The symptoms vary widely depending on which specific mitochondrial function is impaired and which tissues are most affected. They can range from fatal conditions in infancy to later onset problems like blindness, seizures, muscle weakness, or extreme exercise intolerance.
And diagnosis often involves looking at muscle.
Yes, muscle biopsies often reveal characteristic abnormalities like ragged red fibers, which are muscle cells stuffed with aggregates of abnormal mitochondria.
And how are these inherited? You mentioned the mtDNA.
Right, because mitochondria and their mtDNA are inherited almost exclusively from the mother's egg cell, these diseases typically show maternal inheritance. A father with a mitochondrial disease won't pass it on to his children.
Okay. And it can be variable within families.
Highly variable. This is due to heteroplasmy. A cell, and thus a person, can have a mixture of mitochondria, some with mutant mtDNA, some with normal mtDNA. Symptoms usually only appear when the percentage of mutant mtDNA in a critical tissue crosses a certain threshold. So siblings inheriting the same mutation might have vastly different degrees of severity.
And mtDNA mutates easily.
Much more easily than nuclear DNA. Probably because it's right there in the matrix, close to where reactive oxygen species are sometimes generated by the electron transport chain, and it has fewer sophisticated DNA repair systems compared to the nucleus. These mutations tend to accumulate over time, especially in long-lived, non-dividing cells like neurons and muscle cells.
Which links back to Parkinson's disease again.
It certainly reinforces the link. We talked about complex eye dysfunction in PD. The initial clue came from addicts exposed to MPTP, a contaminant that specifically destroyed dopamine-producing neurons by inhibiting complex I. And patients with typical PD often show reduced complex I activity in the affected brain regions. Plus, environmental exposure to things like rotenone, another complex I inhibitor, is a known risk factor.
And what about the connection to aging? Does accumulating mtDNA damage make us age?
That's a long-standing hypothesis, and the evidence is intriguing but still complex. Older individuals definitely have more mtDNA mutations. The big question is cause versus effect. However, experiments with mutator mice engineered to have a faulty mtDNA polymerase that makes lots of errors, are telling.
What happens to them?
They show many signs of premature aging. Weight loss, hair loss, graying, osteoporosis, hearing loss, heart problems, and they have significantly shortened lifespan. It strongly suggests that mtDNA integrity is linked to the aging process, although the exact role in normal human aging is still being worked out.
Fascinating. Okay, what about diseases related to peroxisomes?
Peroxisomal disorders are generally rarer but often severe. The classic example is Zellweger syndrome.
What does that involve?
It's a devastating inherited disease affecting brain development, liver function, vision. Infants usually don't survive past the first year. The underlying problem is fascinating. The peroxisomes themselves often form, but they're essentially empty ghosts.
Empty?
Yeah. The dozens of enzymes that are supposed to be inside the peroxisome are actually synthesized correctly in the cytosol, but they fail to be imported into the peroxisome. They just float around uselessly in the cytoplasm. It's caused by mutations in genes required for this protein import process.
So the delivery system is broken.
Exactly. A failure of protein targeting and import leading to catastrophic consequences.
What an incredible journey we've taken, really, from the ancient bacterial origins of mitochondria through this unbelievably complex dance of electron transport in that spinning ATP synthase.
Right, and even touching on peroxisomes and their unique roles.
And finally, seeing how absolutely critical all this microscopic machinery is for our health and what happens when it goes wrong.
It really does hammer home how fundamental cell biology is. It underpins literally everything from taking a breath to understanding complex diseases like Parkinson's or these inherited disorders.
It's amazing. So maybe a final thought for everyone listening. The next time you take a breath, just pause for a second. Think about that oxygen traveling to trillions of tiny ancient powerhouses inside your cells, driving processes that started billions of years ago.
A direct connection to the deep history of life.
Absolutely.
Yeah.
What does exploring this tiny world inside you make you curious about next? There's always more to discover. Thanks so much for joining us on this deep dive.